Possibilities of gene therapy to prevent autism. I am proposing that fetal gene therapy will eventually be used as a tool to prevent autism, but not at this time because of the complexities and multifactorial causes of the disorder. Genetic testing and gene therapy techniques have the potential to be useful in the early diagnosis, treatment, and prevention of autism in the future. Autism spectrum disorder is a complex neurodevelopmental disorder. It is a heritable disorder and thought to have multifactorial causes, including genetic mutations and environmental factors such as exposure to toxins, illness and infection, and injury of the mother. These gene mutations disrupt mechanisms and pathways of neural development and neuronal communication along with the receptors of neurotransmitters such as oxytocin, serotonin, and glutamate. The characteristics of autism include failure to develop normal social relationships with others, impaired development of communication skills, repetitive movements and behaviors, difficulty with eye contact, learning disabilities, attention difficulties, and behavioral problems. From a brain physiological standpoint, an autistic brain is smaller at birth and then grows rapidly from ages 2 to 3, where it is 10% larger than normal, and then it slows down at adolescence, where it is only 1 to 2% larger. This research will use the neuroligand NLGN gene as the genetic material the vector carries. NLGN encodes cell adhesion molecules that are present at postsynaptic side of the synapse and has been linked to autism. The gene therapy technique uses a virus as a vector to deliver genetic material to human cells in an attempt to alter or correct mutations of genes. For this research, an adenovirus will be used as the vector, carrying an NLGN gene, and it will be injected into the cerebral ventricle. The goal of this research is to develop an effective fetal gene therapy technique to prevent autism. The independent variable is the 2-week intervals for taking images and the 3-month intervals of conducting cognitive tests. The dependent variable is data from the ultrasound, MRI, and fMRI images, along with the results from the cognitive tests. The controlled variable is that 40 participants receive injection of gene therapy vector carrying NLGN gene. 40 participants receive the placebo. There will be 80 pregnant women between the weeks of 18 to 30 who are the participants. They will be from a variety of backgrounds. There will be 8 different groups with 10 participants in each group. 1. Older women 35 and up. 2. Younger women 16 to 34. 3. Twin pregnancy. 4. Single pregnancy. 5. Have gene mutations linked to autism. 6. Have no gene mutations linked to autism. 7. Pregnant with males. And 8. Pregnant with females. To recruit the participants, ads will be placed in newspapers, websites, and social media. There will be compensation and a consent form must be signed. The materials. Genetic screening will be conducted. For the CVS tests that samples placental tissue, a catheter is used. For the carrier test sampling the partner's cheeks cells, a cotton swab will be used. An ultrasound, MRI, and fMRI will be the imaging machines used. The gene therapy vector is an adenovirus, one with the NLGN gene and the other without NLGN gene, the placebo. Amniocentesis uses a hollow needle to get the sample, and cognitive and social tests and observations will include a questionnaire packet and observational studies, which follows autism screening interview guidelines. Participants will be instructed to not change their daily lifestyle, record possible exposures to toxins, illnesses, infections, and injury. When the study starts, they will need to bring their partner in for testing. They will need to come into the lab once every 2 weeks during the pregnancy. After the child is born, they will need to bring the child into the lab for cognitive and social testing every 3 months and for imaging testing twice a year until the age of 18. The procedure: a consent form must be overviewed and signed before the research begins. Genetic screening tests will then be conducted for mutations linked to autism, a CVS test for the participant, a carrier test for the partner. Once those genetic test results come back, the participants will be split into those 8 groups. Participants will undergo imaging tests of their fetus's brain every 2 weeks, starting at the 18th week of pregnancy. Those imaging tests include ultrasound, MRI, and fMRI. Then injection of the gene therapy vector, either carrying the NLGN gene or the placebo, will be injected into the lateral cerebral ventricle at the 20th week of pregnancy. And gene therapy is done by cells are first removed from the patient. In the lab, adenovirus is altered so it can't reproduce. A gene, the NLGN gene, is inserted into the virus. The altered adenovirus is mixed with cells from the patient. Then the cells from the patient become genetically altered. The altered cells are injected back into the patient, and the genetically altered cells produce the desired protein, which is a corrected NLGN protein. An amniocentesis will then be conducted at the 30th week. Cognitive and social testing and observation conducted every 3 months when child is 6 months old to 18 years old. And the imaging test will be twice a year when child is born to 18 years old. The conclusions are gene therapy is a technique that is still in development but shows promise as a possible treatment for autism. The limitations and obstacles of this are that many genes are known and unknown that are linked to causing autisms, and other factors may be involved, so gene therapy may not be the only technique that helps. The advantages is that gene therapy is a targeted, specific treatment for autism.